everyone. Today I'm going to present the article Tazobactin kills bacteria by a two-pronged attack on the cell envelope. This article can be found in Nature in 2022, and I will show it simply by introducing the Tazobactin of discovery, structure, antibacterial mechanism, and its potential in combating antimicrobial resistance. Here is the antibacterial mechanism. Before I introduce the Tazobactin, I will briefly review how antibiotics usually work. There are four major antibacterial mechanisms. First, inhibition of cell wall cytogenesis, preventing bacteria from building a protective barrier. And the second one, disruption of the cell membrane, damaging the microbial envelope. Third, interference with protein synthesis, blocking bacterial growth and reproduction. The last one, interference with nucleic acid synthesis, preventing bacterial replication. And Tazobactin is unique because it mainly employs a dual mechanism that targets both the first one and the second one of cell wall synthesis and the cell membrane. Nowadays, antimicrobial resistance, or we call AMR, is one of the greatest threats to modern medicine. Many antibiotics lose their effectiveness because bacteria get mutated and develop resistance, like we show in this slide. This is why we urgently need new antibiotics with resistance-avoiding mechanisms. And the Tazobactin is one of the most promising candidates to meet this challenge. And the discovery of Tazobactin is in 2015. And this is a peptide-like compound that specifically targets gram-positive bacteria. Its primary target is lipid-2, a crucial precursor in bacterial cell wall synthesis. What makes Tazobactin especially fascinating is that it not only binds lipid-2 but also anchors to the bacterial membrane, creating a two-pronged antibacterial strategy, and which is shown in our slide. Here, I will introduce structure of Tazobactin. Its amino acid sequence contains several uncommon residues. The most notable is induracidin, a rare amino acid found mainly in peptide antibiotics such as Tazobactin. And this amino acid is critical because it stabilizes the binding interface between Tazobactin and bacterial cell wall precursors, which is shown in the right side. And tazobactin can be divided in two parts, the linear part and the cyclic part. And the indurecidin, which is rare in nature, and usually this needs to be seasonless and cannot be found in nature. So when we want to apply this, we may need to season it in lab. And this is the tazobactin antibiotic mechanism. The first mechanism is binding to lipid-2. By sequestering lipid-2, it blocks the senescence of the peptidoglycan layer, which is essential for cell wall integrity. Without a proper cell wall, bacteria become fragile and eventually undergo lysis. The second mechanism is membrane inquiry. After binding, tazobactin molecules aggregate and form anti-beta sheet structures that disrupt membrane organization. This destabilizes the bacterial envelope and causes further cellular damage. And I will introduce how they check these two mechanisms. The first part is the oligomerization and the membrane damage. For the picture E, this is a GUV and with the lipid bilayers. The red part is a lipid-2 with a tracer atto 550, which means its emission the red light. And when we added tazobactin to this GUV, and we can find the aggregate with the lipid-2 and tazobactin. And for the lipid bilayer region, we can find that the membrane has some damage, and compare with the control, which is not added tazobactin. It doesn't have any damage on the membrane. Additionally, we can see the picture F, and we can find when we added tazobactin, the chemical shift of P rise up compared to not added tazobactin. And for the picture G, after we adding tazobactin to the bacteria for 1 minute, 15 minutes, and 45 minutes, we can see the intensity of the fluorescence grow up. And the second part is tetrabenthene crystals lipid into fibrils and use high-speed AFN imaging show that the tetrabenthene organized lipid into fibrillar sheets on the membrane surface. These fibrils deform the lipid bilayer both above and below the surface, which is shown in picture D, demonstrated by surgical high profiles. And over time, the fibrils integrate into the membrane, producing significant distortions. This structural modeling of the membrane contributes directly to bacterial killing. And we can find it, again we can see the distortion for the membrane shown in 3D images for picture E. The next part is eroding residue covers the interface. The MNR studies reveal that tetrabenthene interacts strongly with the pyrophosphate group of lipid 2. These interactions are mediated by the pi cycle backbone and the side chain of eroding residue, which is shown in the picture B and picture C. Spectral analysis confirmed that eroding residue is prominently positioned at the binding interface, making direct contact with mRNAC and other parts of lipid 2, which is shown in picture D. This explains why enduroresidue is indispensable for high affinity binding. Comparative studies between natural tetrabenthene and synthetic analogs highlight the importance of enduroresidue. In natural tetrabenthene, enduroresidue forms specific interactions with mRNAC that stabilize the entire interface, like the left part. By contrast, replacing this residue with leucine creates a much weaker and less defined interaction, resulting in reduced binding affinity. This illustrates why enduroresidue is crucial for tetrabenthene's potency and resistance avoiding properties. And this part this part is about the tetrabenthene membrane interaction. The high-resolution MNR structures reveal how tetrabenthene and lipid 2 form a highly coordinated complex. The backbone protons, the eroding side chain, and the N-terminus of adjacent molecules together coordinate the lipid 2 pyrophosphate group in picture B and generate the hydrogen bonds, reinforcing the stability of the complex of tethylbetene and lipid 2. Molecular simulations show that inner lipid 2 molecules are bound more tightly than outer ones, further enhancing the stability of the oligomeric complex shown in the picture D. This oligomerization also depletes polar lipid head groups, concentrates lipid 2 tails, and ultimately causes membrane distortions, like picture C and picture E. Simulations also reveal that membrane becomes thinner at the site of the complex. Bacterial assays confirm that tethylbetene causes both membrane depolarization and membrane permeabilization. When compared with other antibiotics like niacin, palatistin, and vancomycin, tethylbetene shows a more pronounced ability to disrupt membrane potential and integrity, which is shown in picture H. This dual impact explains its extraordinary antibacterial activity for the tethylbetene. In summary, tethylbetene works by a two-pronged mechanism. First, sequesters lipid 2, blocking peptidic synthesis. And the second part forms supramolecular fibrils, leading to membrane disruption. And this approach is highly effective, resistance avoiding, and selective. And the study provides molecular insight that could guide future antibiotic development. Thank you for your listening.